Production of penicillin, that is penicillin fermentation. Penicillin is the first antibiotic produced on large scale, and it is one of the best antibiotics available today. It is produced on large scale due to impetus of World War II, and that protected many soldiers from the battlefield diseases. Penicillin was the first antibiotic discovered by Alexander Fleming in 1929, and this discovery was an accidental discovery, as we all know. So, briefly, the discovery is like this: that Fleming observed the production of penicillin F from the fungus called as Penicillium notatum. This was further studied by Flore and Chen, the two scientists, and they conducted trials on production of penicillin from Penicillium notatum by using a technique of stationary mat culture technique that was carried out in liquid broth in the flat bottles. Florey, Chain, and Fleming, they shared a Nobel Prize for this vital discovery in 1942. During World War II and for a time thereafter, penicillin producing fungi were studied in order to increase the yields of penicillin. Penicillium chrysogenum was later found to provide greater yields as compared to Penicillium notatum. Nowadays, highly mutant strains of penicillium are used for the large scale production. These strains are mutated for overproduction of penicillin, and they are mutated for no production of any kind of pigment, because pigment formation was one of the interfering substances present in the finished final product. So that disturbed the quality of the product. So therefore, the mutants of penicillium now have been developed, which produces high yield of penicillin and along with absence of production of any water soluble pigment, that is chrysogenin. Penicillin, it is not measured in terms of grams, micrograms, or milligrams. It is always measured in terms of units. So these units are defined as, one of the oldest units of penicillin is defined as, it is an arbitrary unit. Arbitrary means it is decided by ourselves. Arbitrary unit of activity, and it is expressed as Oxford unit. Nowadays it is not used, but see the definition of this Oxford unit of penicillin. It was defined as the amount of penicillin required to prevent Staphylococcus aureus, a gram-positive sensitive bacterium, over a 24 zone diameter zone of inhibition using a cylinder plate method of assay. That means 1 unit of penicillin, Oxford unit of penicillin, is equivalent to that quantity of penicillin which can produce 24 of inhibition zone against Staphylococcus aureus. So this was the measure of only active units of penicillin. Since penicillin is highly sensitive to environmental conditions, so it exists in active form and inactive form. So in order to measure only active molecules of penicillin, it is measured in units. Nowadays, we know that a new unit of penicillin has been defined. So it is called as International Unit of Penicillin. So it is also the measure of activity of penicillin. So it was established as the amount of penicillin equivalent to zero point 0.6 grams micrograms of an international standard of crystalline sodium penicillin G of about 98% of purity. So that is the definition of international unit. So it is 1 unit is equivalent to the 0.6 micrograms of international standard pure penicillin G with 98% purity. So in other words, 1 milligram 1 mg of current international standard sodium penicillin G will contain approximately 1665 units of activity. You can do these calculations on paper also. Therefore, penicillin activity units can easily be converted to weight basis. Now, the structure of penicillin molecule and the precursors. Penicillin is a generic name applied to a group of compounds having the same nucleus and approximately same antibiotic activity against the sensitive microorganisms. So the basic structure of penicillin, it consists of, it differs, means there are different types of penicillins, and these penicillins differ from each other in relation to R group, that is side chain group. Rest of the structure of penicillin is same in all different types of penicillin. So there is a common nucleus and there is a different type of R chain, R side chain in case of different types of penicillins. So the Fleming's original strain, that is Penicillium notatum, from which penicillin was discovered, when this strain was studied, and while studying this strain, the production of penicillin, when the strain was grown in the medium which he used, where precursor were not utilized, so this Penicillium notatum, it produced penicillin F, which is also called as 2-pentenyl penicillin. So whatever the original medium which was used by Fleming, using Penicillium notatum, he observed the production of penicillin F. Penicillium chrysogenum produced penicillin K with smaller amounts of penicillin F. That means this strain is producing different type of penicillin. This strain is producing different type of penicillin. Later, it was discovered while studying that the addition of corn strip liquor to the production medium, it increases the yield of a new type of penicillin called as benzyl penicillin or penicillin G, as it was produced by the same organism, Penicillium chrysogenum. Further studied revealed that corn strip liquor contains phenylalanine and its breakdown products like pentylamine and phenylacetic acid. So presence of these compounds in corn strip liquor, it leads to production of higher yields of penicillin G. This suggested that these compounds acts as a precursor for the production of penicillin G. So that leads to the discovery that penicillin production is a precursor dependent fermentation. And it is also discovered that the synthesis of a particular type of penicillin by fungi may be directed by the addition of specific precursors. So change in the precursors will lead to change in the type of penicillin. This is the structure of penicillin, general structure of penicillin, where we can see that this is the beta lactam ring and this is nucleus. So you can draw this structure on paper right now. So you have to draw this structure first: sulfur CHNCHC. Add COOH here, add two methyl groups here. Then this nitrogen atom is bound to this keto group, that is C double bond O CH, and then there is NH group, CO group, and there is R group. So this R group is side chain group. This is the general structure of penicillin, and each type of penicillin, it differs in terms of this side chain group. Now the properties of penicillin. Understanding properties of penicillin is very important in order to decide the protocol for the recovery of penicillin during the harvesting of broth. So here are the important properties of penicillin. The activity spectrum. Penicillin is a narrow spectrum antibiotic, and it is active against gram-positive bacteria in presence of medium such as blood, pus, and body fluids. It is active against many gram-positive bacteria, then Nocardia, actinomyces, but in general they are not mostly active against gram-negative bacteria, except at higher dosage levels, probably due to the target of penicillin antibiotic is peptidoglycan, and as gram-negative bacteria have low percentage of peptidoglycan, there is a least efficacy of penicillin. The mode of action. It interferes with synthesis of peptidoglycan. Peptidoglycan is a important component of bacterial cell wall, whereas the concentration of peptidoglycan is more in case of gram positive bacteria, and therefore they act as a major target. So synthesis of peptidoglycan, and therefore this penicillin is active against growing cells. So if the cells are not growing, the penicillin is not effective. Only if the cells are growing, the penicillin is effective, as its target is synthesis of peptidoglycan. Toxicity. It is generally regarded as non-toxic to mammals, but in case of higher doses, it may be associated with certain side effects. In many of the people, there may be development of allergic reactions, which may develop in a small percentage of individuals that leads to the development of allergic itching and rashes on skin. Higher doses may be life threatening. Fourth characteristic, there are several different types of penicillins which are closely related in structure and activity against sensitive microorganisms. A common chemical nucleus and differ principally in chemical structure of a side chain. Different penicillins have different pharmacological properties. This is very important. Therefore, the activity of penicillin is dependent on its side chain group. This is how we can justify it. Next characteristic is its physicochemical characteristic. Physicochemical characteristic is very important in order to decide the recovery protocol for penicillin, because penicillin is a highly sensitive antibiotic or compound. Therefore, proper care has to be taken in order to recover penicillin from fermented broth. So let us understand these properties: that it is soluble in water, it is soluble in acetone, amyl alcohol acetate, cyclohexane, ethyl acetate, ethyl alcohol, etc. etc. That means it is solvent extractable. However, its solubility in different solvents it may vary. It is less soluble in benzene, chloroform, and carbon tetrachloride. So these solvents in which penicillin is insoluble, they may be used as a precipitating agents, and these may be used as a solubilizing agents. That is how we decide the strategy for recovery. Stability characteristics: crystalline salt of penicillin, that means if it is present in salt form like potassium salt or sodium salt of penicillin, it can remain stable at low temperatures and are stable over a period of few months. Aqueous solutions of penicillin are unstable but must be stored under refrigeration. Penicillin is most stable in pH range of near to neutrality, that is 6 to 6.5. It is stable over the range of 5.5 to 7.5. Another property is that penicillin antibiotic is highly sensitive to the degradation by an enzyme called as penicillinase enzyme. Penicillinase, it is an extracellular enzyme which is produced by different microorganisms that belongs to group of coliform, most of the bacillus, and few strains of staphylococcus. So these bacteria they produce this penicillinase enzyme, and this penicillinase enzyme it brings about the lysis of penicillin. That means it converts active form of penicillin to an inactive form, and that inactive form is called as penicilloic acid. So action of penicillinase is that this enzyme it hydrolyzes penicillin to penicilloic acid. You need to remember this name. This is how the reaction happens. So most of the penicillin resistant pathogenic bacteria, means these are the pathogenic strains of bacteria against which penicillin is not effective. So these penicillin resistant bacteria like Staphylococcus aureus they produce this enzyme. Therefore, such microbes are the major threats to fermentation industry. That means if the fermentation broth is contaminated with such bacteria, then there will be huge loss due to the degradation of active penicillin molecules. This is the structure of penicillin. Draw the structure of penicillin in your notebook. You can see this pentagon. You have to draw this containing sulfur and nitrogen here. Then two methyl groups. This is carboxyl group here. This is very important. Then there is nitrogen bound to keto group, and then this carbon is bound to NH and CO, and then it is further bound to R group. So there is presence of CONH bond here that you should note. So this penicillin it gets hydrolyzed due to penicillinase. So it breaks this beta lactam ring. So this ring is broken. So here there is a addition of a water molecule, so that this bond between carbon and nitrogen it breaks and making this molecule of penicillin inactive. So there is addition of HOH which is catalyzed by penicillinase, and as I said it leads to the breakdown of this beta lactam ring. So one hydrogen is added here and one hydroxyl group is added here. So COOH NH. This is called as penicilloic acid. That is the action of penicillinase enzyme which is produced by bacteria which show resistance to penicillin. These were the properties of penicillin. Now Let us talk about semi-synthetic penicillins. Whatever penicillins are synthesized by two ways. First way is chemical synthesis, second way is fermentation, and third way is through combination of chemical synthesis and fermentation. So the result of this is called as semi synthetic penicillins. So this is a combination of biological fermentation and chemical synthesis. Such type of penicillins are called as semi-synthetic penicillins. Now see, they are synthesized by a combined process that includes enzymatic conversion of penicillin G to 6-APA. You need to remember this. APA. APA is amino penicillinic acid. Don't get confused between the earlier slide in which we have discussed about degradation of penicillin to penicilloic acid. Here in this case, penicillin is converted to 6-amino penicillinic acid. So it involves conversion of penicillin G to 6-APA followed by the conversion of 6-APA to desirable penicillin. That is the strategy. So this type of synthesis was possible only after the discovery of penicillin acylase enzyme required for the generation of 6-APA. So the concept is that penicillin G is produced by fermentation using Penicillium chrysogenum. Then this penicillin G is treated with some agent so as to produce 6-APA. 6-APA is the core of penicillin nucleus without side chain. That means this agent is responsible for the separation of R group from rest of the core structure of core molecule of penicillin, and that agent that separates or cleaves this R group, side chain group, it is called as penicillin acylase enzyme. So penicillin acylase enzyme is required for conversion of penicillin G to 6-APA. So penicillin acylase enzyme was discovered from different types of microorganisms. So when penicillin is measured by using chemical assay procedure like iodometric assay or hydroxylamine assay, so this chemical assay it will measure penicillin chemically. Penicillin titer means the concentration of penicillin it was found to be greater as compared to the titer measured by biological assay. So what is the difference? Chemical assay will measure inactive penicillin as well as active penicillin, whereas biological assay will measure only active form of penicillin. So the difference of that quantitation was seen when chemical assay was used and when biological assay was used. This leads to the assumption that this penicillin is produced in two forms, active as well as inactive. So later on, analysis of this fermentation broth, it leads to the discovery of 6-APA. So this chemical which was biologically inactive and which was measured by chemical assay, this inactive penicillin that contains only nucleus without a chain, that is R group, this compound is called as 6-amino penicillinic acid. So later a method for the large scale production of 6-APA was developed after the discovery of penicillin acylase enzyme. So it is shown in next slide. So this enzyme, it separates the side chain from penicillin. So various species of bacteria such as Nocardia, Streptomyces and fungi, including few strains of Penicillium chrysogenum also. They are known to produce penicillin acylase enzyme extracellularly. So this enzyme, it can be produced on large scale, it is purified and it can be used for the production of 6-APA. The method for the production of semi-synthetic penicillin. So as it is said that this is a combination of conventional fermentation process that leads to production of penicillin G. So penicillin G is produced separately. Now you have, it is separated, it is harvested and it is partially purified. Now you have partially purified penicillin G. So this partially purified penicillin G, it is mixed with penicillin acylase producer, that is the culture filtrate from penicillin acylase producer. It will serve as a source of penicillin acylase. So these two are then incubated at temperature of 35 degrees Celsius and at pH 7.5 overnight. As a result of incubation, what will happen? This penicillin acylase, it will act on penicillin G and it will separate R group from the nucleus of penicillin and it will also separate the side chain like this. So it will lead to production of 6-APA. So this 6-APA will be then recovered by using this protocol. The pH of the broth is decreased to 4.3. That will lead to the precipitation of 6-APA and this precipitate of 6-APA will be then separated by a convenient method of filtration or centrifugation. Formation of 6-APA is also accompanied by the formation of side chain group. So the side chain group, that is R group, which is liberated due to the action of penicillin acylase on penicillin G, is phenylacetic acid. So this phenylacetic acid, it can be reused as a precursor in the production of penicillin G. That means it can be reused for commercial production of penicillin G, because phenylacetic acid is a precursor, that is the R group for production of penicillin G. This is how 6-APA is produced on large scale. Now let us take an example of the semi-synthetic production synthesis of semi-synthetic penicillin. So first step, that synthesis of 6-APA from penicillin G. Now the reaction occurs like this. You can draw this reaction structure on paper in your notebook as well. This is the common nucleus of penicillin, NHCO, and then there is R group. This R group is CH2 and this is benzene ring. So this is the structure of benzyl penicillin, which is also called as penicillin G. So the action of penicillin acylase, it takes place here. It separates this R group from this 6-APA. Now this structure, which is called as 6-APA. So it happens like this. There is a addition of a water molecule. This is a hydrolysis reaction catalyzed by penicillin acylase. So this will be, okay, this is not the site. This will be the site for the action of penicillin acylase, where the water molecule will be added. that will lead to addition of OH group here and H group here. So it will be COOH and this will be NH2. So now this part is separated, it is cleaved to form uh, this methyl group bound to benzene ring and a carboxyl group. This is called as phenylacetic acid. And this phenylacetic acid, it is a precursor for the synthesis of penicillin G. The another product which is formed is 6-amino penicillinic acid. So here you can see there is NH3 group. So this is how 6-APA is produced by the action of penicillin acylase enzyme. Then second reaction is to use this 6-APA for the formation of new type of penicillin. Now here we'll see the formation of new type of penicillin. So in the next step, this 6-APA is purified, it is separated, and then it is allowed to chemically react with some chemical group which is important for the formation of new type of penicillin. So this is going to be the R group, side chain group. So here we'll take an example of phenylglycine. So phenylglycine, it is mixed with 6-amino penicillinic acid. So here there is going to be a release of one water molecule. So CONH, and there will be a release of a water molecule, and that will lead to this structure will combine with this structure, and that will lead to formation of new type of penicillin. So you can do this, write this on paper in your notebook. So you can see here the conjugation reaction has taken place that leads to release of one water molecule, and here there is conjunction, there is formation of CONH bond. This is called as phenylglycyl penicillin. Phenylglycyl penicillin, or it is also called as ampicillin. And the ampicillin's property is that it is a type of semi-synthetic penicillin which is having a broad spectrum. That means it can kill gram-negative bacteria also, and it is effective against a wide range of bacteria. So that is the advantage of synthesis of semi-synthetic penicillin, that a desirable broad spectrum semi-synthetic penicillin, it can be produced by using this kind of approach.